And Dr. Kale is the Chief Scientific Officer from Genomenon. His talk is going to be about comprehensive identification of gene-disease relationship across the clinical exome through systematic literature review and parallelized evidence curation.
Thank you so much, and thank you for inviting me to speak. I'm going to talk about solving an ambitious problem by bringing scale to the solution. And the flavors of scale that I'm going to be talking about are mixing automated artificial intelligence solutions to gather and index and organize the evidence, and to bring human expertise to this scalable challenge of parallelizing this curation effort. And the substrate for this is to look at gene-disease relationships across the germline clinical exome. As mentioned, I'm a full-time employee of Genomenon. The way that I've architected my talk here is I'm going to begin by talking a little bit about the problem, talk about our approach to that solution in some detail, present some of the results from that approach, and then compare that to existing solutions and draw distinctions, and then end with some implications for the work. So to begin with the problem, this is a problem that's actually been expertly highlighted by my speakers who preceded me here. VUS are a challenge, and they're proliferating or stabilizing, but they're a big challenge. I might have predicted at the beginning of the next generation sequencing revolution that sequencing more patients would lead to a decrease in uncertainty, but that's actually the opposite of what is the case. Some of those VUSs result from the genes being uncertain in their role in causing disease. The plot here is illustrating in the colored bars well-known genes and well-known to how they associate with disease, but in the background there are numerous genes that are represented in terms of how often they're published in the literature, where their role in disease causation is not clear. Many of these, across the many thousands of Mendelian conditions, are being associated with evidence as being disease-causing, and that information and those insights are being continuously published in the literature. You can see that exponentiation here on this plot, and that exponentiation is not slowing. So there's a challenge to harness this evidence to comprehensively understand how these genes are associated with disease and how to bring that to bear to influence clinical genetic diagnostics. Our approach to that solution, to that challenge in our solution is twofold, as I mentioned. On the left side, I'll be talking about our approach from an automated evidence gathering and indexing perspective. And then on the right, I'll be talking about our approach to bringing expert human curation at scale to that challenge. And then at the bottom, there's a numerical summary of some of those results that I'll go into more detail on subsequent slides. So to begin on the left with the automation, we began with a full complement of all of the published literature, clinical and functional studies, comprising 38 million abstracts. We looked further at a prioritized subset of 9.3 million full-text articles that had genetic information. So full text, this is including the supplemental materials as well as figures and tables. From that automated indexing, looking for genes and diseases and the evidence associated with their role in disease causation, we selected about 9,000 of those references for manual review. And each reference was reviewed and the evidence was assessed, and a role in disease causation for more than 6,000 genes was ascribed based on review of those references. You can see there the internal curation infrastructure and the way that we indexed for diseases, phenotypes, genes, and variants according to those different ontologies. That's the automated part of understanding all of that content and organizing that into a curation workflow, which again then on the left was presented to our curators. The team numbers about 30 curators with 3 full-time team leads, and over the course of 6 months, that evidence that had been automatically indexed was reviewed, systematically following standard guidelines, in this case ClinGen guidelines with some slight modifications, but to classify that evidence for gene-disease relationships. That evidence, in large part, fractionated into clinical studies and functional studies, case level and case control for clinical, and for functional models like cell models and mouse models and patient cells and the like. So the result of that setup was that for those 6,400 germline genes, there were a total of about 8,200 distinct germline diseases that they were found to be associated with. There were 11,000 or nearly 11,000 total unique gene-disease relationship associations, or GDRs, and of those, about 6,000 were affirmative. Namely, there was limited or above evidence to suggest that this was a confirmatory affirmative gene-disease relationship. This is a breakdown of some of those, of all of those GDRs, all 6,000 of those GDRs. On the left is a breakdown according to the designations, the quantitation and assessment of the evidence. About a third of those GDRs that had affirmative designations were definitive, 15% were strong, and about half were either moderate or limited. And then if you look over at the other pie chart on the right, you can see the breakdown according to inheritance pattern, with the lion's share of those GDR inheritance patterns being broken out between autosomal recessive and autosomal dominant. This is a further breakdown. This is now describing for each of the genes in the pie chart how many gene-disease relationships there were. So the vast majority of the genes comprising this resultant data set, 70%, had one gene-disease relationship associated with it. But there were a number, as you can see, 19%, 6%, et cetera, that had 2, 3, and more gene-disease relationships associated with them based on the evidence. And that evidence I'm showing in this swarm plot. The data there is representing the number of references that support each of those GDR associations broken out by different evidence categories, definitive, strong, moderate, and limited. So this makes a nice distribution where you can see that the plurality of definitive genes with GDRs had 4 references each to support that designation, but some had 16. Only very few had 1. And then if you move down the ranks there to strong, moderate, and limited, you see a nice stepwise decrease, but still evidence to support the GDR associations. So that was the summary of the results. There's much more to talk about, but that was summary level. We next turned our attention to compare that to a currently known data set of GDRs, and we looked to ClinGen. So what you're seeing in the Venn diagram is a reflection of the work that we produced for our gene-disease relationship curations for Germline versus what's available in ClinGen. So as you may expect, you see a crescent on either side, but a healthy overlap in the middle. So directing your attention to that overlap, there's about 2,100 of those genes that are in common. And if you break those out in terms of how those genes were associated with diseases, there's approximately 80% agreement. And there's approximately 20% disagreement. And I'll be talking about those in subsequent slides. But the gray crescent that you can see on the left reflects genes that are only present in this study. So we're not present in the ClinGen summary data, reflecting value add to this scaled approach of automating the indexing with artificial intelligence techniques and massively parallelizing the curation efforts. So I'll be talking about those 2 aspects here on the subsequent 4 slides. First, speaking to the unique genes and the GDRs that resulted, you can see here on the left a breakdown of those genes and the strongest GDR level of evidence for each of those genes. Approximately 20% of those genes had at least a definitive GDR. Another 20% had at least a strong GDR, et cetera, et cetera. And then the Sankey plot is further breaking those out for genes for which there were multiple GDRs. So the takeaway from this is that these genes are unique to the present study, but they're nevertheless definitive, strong. They have evidence to associate them with genes, and nevertheless they're unique to this study and not found in ClinGen. Here's an example. The gene is MPC1 and the disease is pyruvate carrier deficiency. At the time of this analysis, ClinGen had not analyzed this. When we performed this work, there had been a study with functional and clinical evidence that was evaluated on September 30, 2023, that's indicated that this was a definitive association. And the evidence there is presented from that publication. So now turning our attention to the middle of that Venn diagram where there's overlap between the genes. This is a Sankey plot beginning with the total, in the middle talking about the way that the evidence fractionated for GDRs for ClinGen, and then on the left talking about how that further broke down for the results for GDRs and evidence categories for the present study. So in text form there, I'm summarizing that there's about half of these genes had simple agreement, meaning there was one GDR and that had a definitive classification, and there was a perfect agreement with ClinGen in the present study. There was an additional 25 where there was agreement, but there were additional GDRs for that gene found in this present study, and a small fraction where the converse was true, where there were additional GDRs found in the ClinGen work. So there's large agreement with the added addition of several GDRs, depending on the genes that you look at. And then in terms of the disagreement, you'll notice there that about 17% have either simple or complex disagreement, but in the analysis, those GDRs were promoted in the present study based on a surfeit of evidence and a scalability of curation to stay abreast of those newly published references. And here's an example of one of those where the classification was designated moderate by ClinGen for ATP1A1 and Charcot-Marie-Tooth, whereas it was classified as definitive in part based on a newly published study, again, that shows both clinical and functional evidence from 2023 that indicated in our analysis, according to those frameworks, that this was in fact now a definitive GDR classification. So the last part of my talk, I'm gonna talk about comparison to the GenCC initiative, which is an aggregation of this collected GDR data across the fruits of 12 different groups having submitted this information. So this is a busy table, but it's really meant to say that we all basically followed the same formula. We looked for the same evidence, we assessed it in basically the same way. We did a comparison to the data there as well. It's important to note here when you're looking at the crescent for GenCC alone that includes somatic level genes as they are associated with somatic diseases, which we did not include in our study. So those weren't removed from this comparison, and the vast majority of those 1,300 genes only in GenCC are likely to have somatic associations. You can see a further crescent of genes that are unique to this present study not found in GenCC, and a further overlap of agreement of about, in this case, 90% agreement. So when we looked further at the genes and the GDRs that comprise the GenCC data set, there were about 5,000 total genes that were present. And when we looked deeper, we found that 20% of those were supplied by a single submitter out of the 12. So there's a fifth of them are unique and only submitted once. Of those 5,000, about half of them had disagreeing evidence classifications for specific GDRs, which is acceptable when you consider that they're fairly meaningless or not impactful or actionable differences, say, between a strong and a moderate. So if you group the classifications, as you can see here, into strong, moderate, and definitive, that's group 1, and limited, disputed, refuted, et cetera, into group 2, there's nevertheless about 15% across the GenCC submissions where there's disagreement between individual submissions across those two groups, suggesting that it's much more of an impactful and meaningful difference. So I bring that up not to disparage GenCC, but to say this is complicated, there's a lot of evidence at play, there's subjectivity to the interpretation, and you might actually expect that there's that level of disagreement. And so when we compared our results to the internal comparison of different GenCC submitters, we see a lot of parity in the level of disagreement and the level of different classifications based on evidence or based on the subjectivity of that interpretation. But the summary here from our comparison with GenCC is that about 90% of the genes in unison here in the present study in GenCC agreed. That's a substantial level of agreement. I mentioned that GenCC internally disagrees 14% of the time, and that just so happens to be nearly the exact level of disagreement between the present study and GenCC. So I would say that that's an acceptable level of disagreement. And I'll make the case here that, as is my want, that evidence should prevail. Evidence should rule the day. There can be disagreement in the subjectivity of the interpretation, but the evidence should be the go-to for resolving those disputes. So briefly here in summary, the study that I presented reflects an ability to curate the germline exome for gene-disease relationships using a combination of AI techniques, most notably here natural language processing, to organize and annotate that evidence, pairing that with expert, human-level, massively parallelized manual curation of that evidence. As I mentioned, this was an ambitious undertaking, and scale was needed to solve this challenge in such a timeline. When we looked at the validity, the quality of those results, we compared that to ClinGen and GenCC and saw a substantial amount of parity, with the caveat that there's additional information, additional evidence, additional genes that were provided by the analysis in the present study. So lastly, I'll conclude by saying our view is that this represents a powerful proof of concept that focused curation efforts coupled with that semi-automated approach, big data approach, automated indexing to massively parallelize this evidence curation is fit to purpose to solve these ambitious challenges. So thank you for your attention, and I'm happy to take any questions.
Excellent. So the 25% additional GDRs that you found in your study, what proportion of that could have been because ClinGen lumped the phenotypes? Did you actually follow a similar lumping splitting criteria? Because clinical labs in GenCC, I noticed they tend to go more granular. They go phenotype by phenotype in OMIM, and ClinGen doesn't. So could it have been?
It's not, I think the question could be heard because you're Mike, but it's not exclusively because of that. I'm very well aware of that challenge. Whenever you're cross-comparing these two data sets, one of the major challenges is reconciling this disease ontology and that disease ontology. So the numbers that I showed reflected a subset analysis, but it was a substantial subset. It was at least 1,400 for both ClinGen comparisons and GenCC. Unfortunately, I don't know exactly the breakdown, but we were mindful of that when we were doing the comparison.
It would be interesting to look at your 25% and
sure, I agree.
Proportion.
Yep, so this work is in the process of being written up.
Great, thanks for a good talk. Marina DeStefano, the Broad Institute, creator of the GenCC database. So it's great to see someone, use the work that we have. So comments and questions.
Sure.
So first comment, we'd love to chat with you about submitting to our database because gene curation is so hard and it takes so much time to do it. And so when public can use it, we'd love to chat about that.
Sure.
Second comment, there are no somatic submissions in the database unless Orphanet has submitted them as supportive. It's all monogenic gene disease relationships. So check into that. I guess my question is then, what ontology do you use? Because it is such a difficult thing and not a standardized across the field usage.
Yep, so we have two on the go here for our index data and for our manual curation. Those two are Mesh and Mondo that we're using. And as I mentioned in answer to the other question, we're sensitive to the difficulty in reconciling those, so that was largely a post hoc manual process.
Yeah, great.
But in response to your first comment, I would welcome the opportunity to chat.
Okay, great. Thanks.
Great discussion. Thank you for a great talk.